Hi and welcome back to our next lesson. Following on from last lesson, the answers on the screen here at the moment are the questions that you should have answered at the completion of last lesson. So let's just go through our answers here just to ensure that you're on the right track. Mark them correct if they're correct, mark them incorrect, and fix them if they're not. So distinguish between stoma, stomata, guard cells and lenticels. Our stoma is just one individual pore in the underside of the leaf where water and gases can pass through there. Water in the form of water vapor can pass through there. Our stomates or stomata are plural, so that is many of the stoma that we would have in our leaf. Looking at our guard cells are the two bean-shaped cells that surround the stomata. So the stomata being the pore, the hole, the guard cells being the cells around the outside, and they open and close to allow that exchange of gases. Then we have our lenticels. They are the pores in the woody tissue of the plant, again allowing that exchange to occur. So what gases are exchanged through the stomata? The gases that are exchanged through the stomata are our carbon dioxide and our oxygen, but also water vapor. So describe the changes that occur in guard cells that cause the opening and closing. So when guard cells are full with water, they become turgid, and when they're turgid, the thin outer walls stretch, whereas the thicker inner walls don't move so much. And then what happens is they open. When they lose water, they then will close and to prevent that exchange occurring. Number 4, identify the origin and outline the use of each of the three gases that move in and out of the cell. So we first of all said we have oxygen that's made inside the leaf via photosynthesis, and it's then used in respiration to provide energy for the cell. We then have carbon dioxide. It's taken in from outside the plant, so from the air into the plant, and is used in photosynthesis to provide glucose for the plant. Then we have water, which the water vapor actually comes from within inside the plant. So the water comes from outside the plant into the roots, then moving up through to the leaves, and then water vapor is formed then and moves out of the cell. Now describe the conditions that affect the opening and closing of the stomata. First of all, we've got temperature increases. So if temperature increases, more water is lost and the guard cells may close. We have, if water availability decreases, carbon dioxide levels in the plant might increase. If carbon dioxide levels in the plant increase, the stomata will close to reduce any more carbon dioxide coming in. And obviously the reverse is true. If the carbon dioxide levels are too low, they will open to allow more to come in. We also have light. Light will cause the stomata to open. Photosynthesis occurs during the day, so that light will stimulate that. And then also humidity. If humidity is high, our transpiration, our loss of water is released. So fix up your answers. Moving on now to today's lesson, which is on page 36 in your book, and we're looking at the transport system in plants. So you've got a big inquiry question at the top. Why do plants need transport systems? So I want you to have a bit of a think about that. But like humans and other animals, we have transport systems within our body in the form of our veins, our arteries, our capillaries to move those nutrients and the gases throughout our body that we need. So too do plants. In this case, plants have two transport systems in the form of phloem and xylem, and again to allow the plant to transport the nutrients that it needs up and down the plant. So in plants, the transport system is used to transport minerals, water and sugar. So we call these vascular bundles, and in the vascular bundles it's made up of our xylem and our phloem. And if you can see over here in our diagram here, you can see that the water is coming in and moving up to the top of the plant there. Whereas in the case of our phloem, our food is produced in our leaves. They photosynthesize there, and then it needs to move up to the flowers and the leaves that might be or the stems at the top, but also down to the bottom. So it's a two-way system there. So looking now at our xylem is specialized for the transport of water and dissolved minerals from the roots to the leaves. It's a one-directional movement only. So what's happening is there's a pull of water drawing the water as water is lost through the leaves. So plants go through the process of transpiration where water is lost through our leaves and through those pores in the form of water vapor. So as our water is being pulled out through the leaves, it draws more water up. So there's that constant pull, and that's then also drawing more water in from our roots. Phloem, on the other hand, is also a specialized tissue, and it's specialized and designed to transport larger molecules around, such as glucose. It is a two-way process, and the reason for that is this food is being made in the leaves, but the stem and the flowers further up the plant will also need that food, as will the leaves, flowers, root systems down in the bottom. So it's transporting those sugars to all parts of the plants that need it. So what you're going to do today, and unfortunately you're not in the lab so you can't, but you can have a bit of a go at home if you've got some of the materials, is a celery prac. So what our plan was to investigate the direction and the structures through which water and dissolved minerals will move through a celery stem. So a celery stem, pop it into a red food dye for a couple of hours, and then having a look at what happens. I want you to have a go at filling out what possible hazards there are and how we can manage those with doing this prac. So remove the celery from the solution. We're going to have a look at the spread of the dye. Where's it gone? Then we're going to do a transect. A transect is going across the stalk and having a look at where that dye is located. So if we have a look in this picture here, you'll notice that it sits in this solution for a bit and water has moved through, and you'll notice that the red will be around the outside. You can do this at home if you do have a celery stalk with some leaves on the end, but you can even give it a go with just a flower cut out of your garden. So then what you're going to do also is cut a longitudinal section. So that's a slice downwards of the celery stalk. And again, have a look at those tubes that have got the coloured water. And then we're going to draw some pictures. Now, because you can't do this yourself unless you've got celery, there are a couple of videos on Compass that I'd like you to watch demonstrating this happening so that you can still do the rest of the prac. But if we have a look here, the one to the left here is your transect. And this one is your longitudinal. So you can see here that the dye has moved up, and we can see that red dye in the parts of the plant. That is where the water has moved up through the xylem. And if you look on the right hand side here, you can see that those lines go the whole length of that celery stem. So what I'd like you to do is watch the couple of videos that are links on Compass and watch those. That'll be as if you were doing the prac yourself. One is of someone explaining that prac. Another one is showing how they make that transect as if you were doing it yourself, just using a different type of stem. And then there are some, another one again on our celery, so that you can answer all of those questions that are required of you, which will be on page 39 in your booklet. So what I will expect from today's work is seeing your diagrams on page 38, you can draw them there, and your questions on page 39 to be uploaded to Compass so that I can see that you're staying in touch with where we're at. I'll talk to you soon. Bye-bye.